My name is Ostenamo, and I'm here to present a topic of expression of Brassinosteroid in bacterial cells for functional studies. I'll start off by talking about the main aim of the project. The main aim of the project is to express Brassinosteroid in bacterial cells for functional studies. Now let's talk about the function of Brassinosteroids. They are a class of plant hormones that play a crucial role in various physiological processes, including cell elongation, cell division, and vascular differentiation, and is responsible for environmental stresses. So, brassinosteroid receptors in vascular plant tissues confer drought resistance without negatively impacting their growth. And this project aims to elucidate its functions. And now to our main slide, which is the strategy for the vector insertion and bacterial transformation. So this stage talks about a lot from the process of choosing a vector to the process where we transform our competent cells. So we are going to start off by talking about why we are using bacterial cell transformation. We are using bacterial cell transformation because they multiply rapidly, and then also we are not going to need post-translational modification. That is why we are using bacterial cell modification. So we start by acquiring our vector. In this case, we are using PMDC32 vector from a reliable source. And then we move on to the stage where we choose our restriction enzymes. So in this case, we are going to consider EcoR1. The next stage is to design the primers for gene amplification. So we will design a PCR primers to amplify our gene of interest. The primers should contain sequences that are complementary to the ends of the linearized vector to facilitate cloning. The next step here is to amplify the gene of interest. So we perform a PCR to amplify our gene of interest using the design primers, and we will use a high fidelity DNA polymerase to minimize errors. The next step is to digest the PCR product and the vector. So we digest both the PCR product and the linearized PMDC32 vector using the choosing restriction enzymes, and then run the digested product on the agarose gel to verify successful digestion. The next step again is to purify the digested PCR products. So we purify the digested PCR product using a gel extraction kit to remove excess primers and nucleotides and enzymes. The next step is ligate gene into the vector. So we perform a ligation reaction to insert the purified PCR product into the linearized PMDC32 vector and use DNA ligate to covalently join the ends of the vector and the insert. The next step here in our slide is to transform competent cells. So we transform chemically competent agrobacterium tumefaciens cells with ligated plasmid and plate the transformed cells on selective agar containing hygromycin and kanamycin, because these are a selection marker present in our vector PMDC32. From our main slide, we go to the next slide, which is vector validation selection. So we will screen the transformed colonies by conducting PCR or plasmid isolation, followed by restriction digestion to confirm the presence of the insect, and then sequence the plasmid from positive colonies to confirm the correct insertion of the gene of interest. Now on to bacterial cell transformation. So we prepare the agrobacterium plant for transformation. So we transform agrobacterium tumefaciens with a confirmed PMDCR32 plasmid and perform plant transformation using the agrobacterium mediated method suitable for the plant species. So in this case, we are considering Arabidopsis thaliana. So we are going to use the floral dip for Arabidopsis thaliana. And then we will select and screen the transgenic plant. So we are going to select and screen the transgenic plant based on the presence of the selectable marker and confirm the stable integration of the gene of interest by PCR or other molecular methods. Now, for us to know the expression, we are going to perform expression analysis. We will confirm the expression of the gene in transgenic plants using techniques such as quantitative real-time PCR or Western blotting. In the next step, we are going to do functional studies. So we conduct functional studies based on the goals of our research observing phenotypic changes or analyzing chemical parameters. And then we analyze our data and draw conclusions based on our experimental results. Thank you.